This is Bio 182, Lecture 3 on macroevolution. So far we have looked at microevolutionary processes. That would be when we talked about natural selection, gene flow, genetic drift, mutations, those kinds of things. So we were talking about changes in allele frequencies in populations over time and how adaptations arise through natural selection. But what about the big picture in all of this? So that's where macroevolution comes in. So when we're talking about macroevolution, we're talking about the origin of new species, which is speciation. It can also be the origin of new genera, families, classes, orders, phyla, or even kingdoms. So we're going to answer a few different questions here. We're going to discuss what is a species and look at different patterns of speciation. So first, to define what a species is a group of organisms that in nature can produce viable and fertile offspring. This is according to the biological species concept. There's a few different definitions of species. This is the one that we are going to use. And so there's different parts of this definition that are important. So what we need to focus on is nature, viable, and fertile. When we're talking about the creation of a new species, a species has to be something that could occur in nature. It would produce viable, meaning that they would survive. So it has to produce offspring that would survive, and fertile, meaning that they would reproduce. So the organism would have to be able to occur in nature, and then it would have to be able to create offspring that would survive and reproduce. So when we're thinking about things that we could sort of mix this different species and create new species, one thing that students often ask about is, what about a liger, right? A lion and a tiger combined. And so lions and tigers can technically reproduce and create a viable offspring. Whether or not it's fertile is a different story. But we could create something in an artificial or a zoo environment where you have a lion and a tiger combining to make a liger. However, those two species would never, under natural circumstances, be in the same place at the same time and reproduce for various reasons. And so a liger would not be a species according to this definition because it wouldn't occur in nature. There are millions and millions of different species of plants and animals, and if we also think about bacteria and fungi and things like that, we are many, many, many millions of species on the planet. Even when we think about different species of individual things, right? When we talk about something like ants, we just say, oh, I have ants in my backyard. I have ants crawling on my kitchen counter, things like that. In reality, there are more than 20,000 species of ants, and there's 500 or so in Arizona alone. And so we're going to talk more about the different processes and patterns that create different species, species that look completely different from each other, or species of something like an ant where we sort of lump them into the same category, but in reality there's tens of thousands of different species of those. So when we're talking about different patterns of speciation, there are two different possible ways that this can happen. The first one is called anagenesis or phyletic evolution. And this is where you have one species and then you accumulate changes through those microevolutionary processes like natural selection and mutation and things like that. And those processes can lead to a change in that population or species that creates a new species that is completely different from the first species. So you start with one species and that species evolves into a completely different species over time. So that one is usually pretty easy to understand. We have one species, it goes through changes, its environment selects for certain things, and we end up with a completely different species that would, if they occurred together, be unable to reproduce with that original species. The other option is called cladogenesis, which is branching evolution. So in this case, we start with one species, and then we have a new species that branches off from that species, and both species occur in the same time. And so you are increasing the number of species through this process. Whereas with anagenesis, you were starting with one species and it was evolving into a different species. In this case, we have a species and a new species branches off from it. So when we're talking about how this can happen, how we can create new species, it basically happens through reproductive isolation. We're going to talk about several different types of reproductive isolation, but in general, what reproductive isolation means is that we have an original population, and through time, you have one or two or three, however many new populations that have differences for some reason. And there's something that's going to keep those new populations from mating with each other. So we're going to have new populations that are reproductively isolated from each other and possibly from that original population. So what does reproductive isolation entail? We have various types of reproductive isolation we're going to talk about. And those can be either prezygotic barriers that prevent reproductive isolation or postzygotic. So prezygotic means that they're going to impede the mating between species or hinder fertilization if mating is attempted. So these are going to be things that occur before you even create an offspring. So they either occur before mating or shortly after. So they're things that include like habitat, behaviors, or temporal issues, or issues once mating is attempted or occurs that makes it so that that does not result in any kind of offspring. We're going to talk about those a little bit more in detail. We're going to start with those prezygotic barriers, meaning that it's going to happen before an offspring is actually created. So the first one is a prezygotic barrier. It's called habitat isolation. And in this instance, we have two species that live in different habitats in the same area, and they rarely meet. So in this case, we have species of spiders. If one of them lives on the trunk of a tree and one of them lives on the upper canopy of a tree, they live in the same area. Hypothetically, they could interact with each other, but they don't prefer the same habitats, and therefore they will not interact and will not mate because they have different habitats that they prefer. Another example of habitat isolation is in garter snakes. There are terrestrial garter snakes and aquatic garter snakes. Because they live in different habitats within their environment, they do not run into each other, and therefore they don't mate with each other. And they've created different species, one that prefers terrestrial environments and one that prefers aquatic environments. If you put these two species in a lab situation together, they will mate with each other, and they can, but under natural conditions, they would avoid each other because they don't live in the same area. And therefore, you have garter snakes that are terrestrial and prefer a terrestrial habitat, and garter snakes that are aquatic and prefer an aquatic habitat. The second type of prezygotic barrier is called behavioral isolation. In behavioral isolation, you have species that do not mate with each other because they have different courtship rituals. In this case, we have what are called lacewings. Lacewings are carnivorous. They eat other pests, and so they're good to have around. They all look very similar. All species of lacewings look very similar, but they have different courtship songs. So when a male and a female lacewing are trying to mate with each other, they will sing a song, and if that song doesn't match, they won't mate with each other. So you have to be in a mating situation with a lacewing of the same species. Otherwise, a different species has a different song. And if you look at the bottom of the slide, you have 5 different types of lacewings, 5 different species of lacewings, and their songs, right? And so they look different from each other. And so lacewings will not mate with another species because they don't have the right song. Another example of this would be a lot of types of birds. Some birds, like birds of paradise, have very intricate dance performances that the male puts on for the female. And if the male is trying to mate with a female of a different species, the female is not impressed by a different species courtship dance. She's only impressed by the courtship dance of her males of her own species, and therefore females will not mate with males of a different species because that courtship ritual doesn't match up. So they have behavioral issues where they're not matching up, and therefore they will separate into different species because they will only mate with males that they have a preference for that courtship display. Another type of prezygotic barrier that can create different species is called temporal isolation, meaning that the species breed at different times. This could be a different time of day, a different season, or even in different years. One example of this is in skunks in North America. There are western spotted skunks and eastern spotted skunks, and there are parts of their ranges that overlap. However, western spotted skunks mate in late summer, and eastern spotted skunks mate in late winter. And because of that, they are not ready to mate. They're not fertile at the same time of the year, and therefore they're unable to mate. And this makes sense when you think about the winters in the eastern half of the country, where during the winter you would not want an eastern spotted skunk to have babies that are born in the middle of the winter when there is no food around. So instead, the eastern spotted skunks mate in late winter, and then they have their babies in late spring when food is abundant and they can survive. So again, temporal isolation just means that you have species that are breeding at different times, and therefore they are not able to breed with each other because they're not fertile and looking for mates at the same time. The 4th type of prezygotic barrier actually occurs after you've gotten over all of the previous hurdles. So you live in the same habitat, the behaviors are not an issue, you mate at the same time of year, so you go to mate with someone who's a different species and you have an issue where your anatomy does not meet up with each other. So you have a tab A, which should be inserted into slot B, and the structures don't match up, and so mating is not able to happen. This happens often in insects. So male insects have penises that are shaped in different ways, and females have vaginas that are shaped also in certain ways, and only males from the same species have the genitalia that will fit into the female's genitalia and allow copulation. So for instance, in June bugs, they have their genitalia that has specific shapes that allows them to fit together. And if a different species were to try to mate with a female or even a male, their genitalia will not fit together and will not allow mating to occur. Mechanical isolation can also occur in flowers. And so within flowers, the nectar and honey and pollen are located in specific parts of the flower, and the pollinators that pollinate those particular types of flowers will only be able to access the nectar and the pollen and whatnot from that specific flower. And if they go to pollinate something that is a different type of flower from the original, the pollen will not rub off in the correct way and will not fertilize the flower. So the last type of prezygotic barrier is called gametic isolation. And this is where species gametes are just incompatible. So we've gotten past all of the other prezygotic barriers. We've gotten past the fact that you don't live in the same habitat. We live in the same habitat. Our behaviors align. We're mating at the same time of year. Our genitalia fit together. So now we're able to copulate. But once we get to the point of a sperm trying to fertilize an egg, the gametes are not able to recognize each other and they're not able to fertilize. So on the surface of sperm and eggs, there are signaling molecules or proteins, and it's sort of a lock and key situation where the sperm has to have the correct key to enter the egg. If you have gametes from two different species, the egg will not recognize the proteins on the surface of the sperm, and that sperm will not be able to fertilize that egg. And so in that way, we can have gametic isolation where you are not able to reproduce with the same species or with a different species because your gametes are so different that they're unable to fertilize each other. That type of a reproductive barrier, so the gametic isolation, is very important for things that have external fertilization. So something like a sea urchin, which just releases sperm into the water, and the sperm float over and fertilize neighboring female sea urchins, or pollen from trees that use the air to float their pollen to other plants instead of using pollinators. It's a very similar thing where they have specific proteins on the surface of their sperm, and those sperm have to match up correctly with the eggs of the females of their species, and if they don't, then fertilization does not occur. Okay, so we've talked about the prezygotic barriers. Now there's a couple different types of what we call postzygotic barriers, meaning that we've gotten past every single one of those prezygotic barriers. We live in the same habitat, our behaviors match up, we mate at the same time of year, our genitalia is able to copulate together, and our gametes can fertilize each other. We've gotten past all that. And so what else can happen are called postzygotic barriers, which are things that happen after the gametes have fused and created offspring or potential offspring. So the first type of postzygotic barrier is called reduced hybrid viability. And this is where hybrids will fail to develop or reach sexual maturity. So we have two species of toads in this situation, both of the genus Rana. And what happens is if we have a male from one species and a female from the other get together and mate, their eggs will fail to develop. During development, there's a lot of different genes that get turned on, and you have to have everything sort of match up to create a viable offspring. And if the timing of development is not correct, the eggs in this instance will just not develop and no offspring will come from them. So the hybrid between two species is not viable. And so that's why we call it reduced hybrid viability. Another type of postzygotic barrier is called reduced hybrid fertility, which is where you have the parents are able to get together and they create an offspring that actually survives the embryonic stage and becomes an actual living adult or juvenile of their species. A very well-known example of this is where we have the offspring of a horse and a donkey, and they create a mule. The problem here is that horses have 64 chromosomes and donkeys have 62. And when they create a mule, they have 63. They have 63 chromosomes. And therefore, when that mule goes through the process of meiosis to create gametes, it's going to have a difficult time, and there's going to be issues with creating gametes that have an appropriate number of chromosomes to be able to then make their own viable offspring, because they have an odd number of chromosomes, and especially in comparison to their parents, where their chromosomes are different from either of the parents. Something similar actually happens in humans and chimpanzees, where humans and chimpanzees have almost all of their DNA is identical, but humans have 23 pairs of chromosomes, so 46 chromosomes. Chimpanzees have 24 pairs of chromosomes, so 48 chromosomes total. And so what seems to have happened in humans is that one of those chromosomes that chimpanzees have fused into a single chromosome. So two chromosomes from chimpanzees fused into one chromosome in humans. And so we had a reduction in the number of chromosomes. And therefore, that's one of the reasons that humans can't create a viable offspring with a chimpanzee. The third type of postzygotic barrier is known as hybrid breakdown. And this is where the F1 generation of a hybrid between two species is viable and fertile and is able to survive and reproduce. But then when those individuals from the F1 generation start creating the F2 generation, those F2 offspring of the hybrids have reduced viability or fertility as a result of gene incompatibility. And so this happens often in cichlid fish, which occur in Africa, where if you have two species of cichlids that create a hybrid, the F1 generation does fine. But once you get past that, the subsequent generations of offspring are not viable or fertile. So we've talked about 8 different types of reproductive isolation. And so what can actually happen to initiate that to occur in species? What actually is the cause of reproductive isolation? Where does that reproductive isolation come from? So there are 2 modes of speciation. The first one is called allopatric speciation, which is where there is geographic isolation between 2 groups that leads to them diverging enough to be considered different species. And there's also sympatric speciation, where there's no geographic isolation, and it's an alternative mechanism, but there are alternative mechanisms for isolation. So allopatric speciation is where you have a geographic barrier that is occurring between two groups, and over time they become 2 different species. So you have an original population, something happens where there's a new geographic barrier, the 2 populations are split into 2 different areas, they adapt and change and become well suited to their specific environments, and over time they become different species. One really well-known example of this is occurs in shrimp that occur near North and South America. So millions and millions of years ago, North and South America were not joined together. Over time, we formed the land bridge between North and South America. We think that was about 3 million years ago. There's shrimp that live in that area, and when that land bridge formed, there were 2 distinct populations that were created: the shrimp that live in the Atlantic side and the shrimp that live on the Pacific side. And it just so happens through chance that shrimp in the Atlantic have a right pincher that is enlarged, and shrimp that live in the Pacific have a left pincher that is enlarged. They used to be conjoined into one population, but it just by chance, we ended up with more individuals with an enlarged right pincher in the Atlantic side and more with a left pincher on the Pacific side. And over time, they've become two distinct species, but we know that they used to be conjoined into one species, and it was separated by the joining of North and South America. So that geographic barrier caused them to create two separate species because there's no longer intermixing of genes between the two populations, and they have now evolved to be more specific to their own environments. Allopatric speciation can also lead to what we call adaptive radiation. So this is where we have the evolution of many diverse adapted species from one common ancestor, and this is very common in island systems. So assume that we have a mainland population, and over time, some of the individuals from the mainland population make it over to an island, and then create a new species, and then that species hops over to another island and another island and another island, and over time we have new species being created according to the specific environments on those different islands. And so we call this adaptive radiation, where we have an initial species and then we have many species radiating outwards from that initial species. There's quite a few examples of adaptive radiation. One of them is marsupial radiation in Australia. Marsupials are mammals that have a pouch. There was an original marsupial that colonized Australia. And from that original marsupial, there was a radiation event where we had many, many different marsupials evolve from the original marsupial. We also see it in Drosophila, which are fruit flies that live on the Hawaiian Islands, where a fruit fly was able to get to one of the Hawaiian Islands and then gradually moved in a linear fashion from one Hawaiian island to the next, and creates new species that are adapted to the specific environments as you go along. We also see this in Darwin's finches. We talked about those last time, as well as Hawaiian honey creepers. We also see adaptive radiation. So how big does a barrier have to be to allow allopatric speciation to occur? That's going to depend on the organism. So when we think about something like the Grand Canyon, an organism that can fly won't necessarily find the Grand Canyon to be much of a barrier. Something that is small and lives on the ground would find the Grand Canyon to be a barrier. So we actually have different species of squirrels, ground squirrels, things like that, that live on the south rim of the Grand Canyon compared to the north rim, because skittering down one side of the canyon, crossing the river, and skittering up the other side is quite a feat for something this small to make it that far. And so over time, the species on the south rim of the Grand Canyon and the species on the north rim of the Grand Canyon have diverged into separate species. We also see what we call ring species, which are a connected series of neighboring populations, each of which can interbreed with closely sited related populations, but for which there exist at least two end populations in the series, which are too distantly related to interbreed, even though there's a lot of gene flow between the linked populations. So in this case, we see this in the Ensatina salamanders in California, where we have species. So if we look at the figure towards the left-hand side of your screen, we start with the pink species. The pink species and then purple, blue, teal, and green. Pink can breed with purple, purple can breed with blue, blue can breed with teal, teal can breed with green. But if we were to loop those species around, pink and green are two differently related, or two different from each other. They're too distantly related to be able to breed with each other. So you can breed with the species that is nearby you, but you can't breed with species that are very far away from you. And so some of these individuals may exist in the same region, which is sympatry, and it closes the ring of these species where you might live in the same area and could potentially interbreed, but because you're so distantly related from the original species, you're not able to, once you get to the end of this line of speciation events, that the most recently speciated species is so different from the original that there's interbreeding in the middle, but the two ends cannot breed with each other. Ring species are important because they do represent speciation and are evidence of evolution. They illustrate what happens over time as populations genetically diverge, specifically because they represent in living populations what normally happens over time between long deceased ancestor populations and living populations, in which the intermediates have become extinct. The evolutionary biologist Richard Dawkins remarks that ring species are only showing us in the spatial dimension something that must always happen in the time dimension. So we're able to see through a group of living species something that normally occurs over millions and millions of years, but we're able to see it in the same time frame, just in different spatial areas. So a lot of the time we think of allopatric speciation as occurring across a very large barrier. The barrier can be quite small. There's a lot of species of butterflies that are endemic to South America, and there are very small barriers within that continent that create different species of butterflies that are not able to reproduce with each other. And so it's not necessarily something huge like an ocean or a mountain range. It can be something small, something like a river, or something even smaller, depending on the species and its ability to move. The second type of speciation that can occur is called sympatric speciation. And this is where there's no geographic isolation at all that is causing species to diverge from each other and create new species. Sympatric speciation can be caused by sexual selection or polyploidy, and we'll talk a little bit about both of those. But in this case, we have an original population that all lives together. There's no geographic barriers, but within that population, we end up with new species occurring. So in this example, we start out with males with color variations, and those variations are genetically determined. At first, females are going to breed randomly, so the females don't show any preference for males. But over time, we're going to possibly see females show a preference. In this case, we have some females preferring blue males and some preferring green males. And so what will happen is that over time, we will have male offspring that will inherit blue color, and female offspring will inherit the preference for blue males. And so when you have that occurring, that group can create its own species, and the group that was preferring the green males will possibly create their own species, where you have green male offspring and female offspring that inherit the preference for green males and create two distinct populations. So males with two color variations, when the variations are genetically determined, will, over generations, you'll have the populations continue to diverge and result in two different species, where you have preference for blue color and females preferring blue color, and then some that prefer a different color or have no preference. We can actually see this in cichlids. So in normal light, female Pundamilia mate only with male Pundamilia, and female Nairi only mate with male Nairi. But in monochromatic light, the color differences between the species go away, females can't tell the difference and will mate indiscriminately. And so that preference for mate is based on color. And so if the water were to become murky or shaded and you weren't able to see the difference, or the females weren't able to see the difference between the males, you might actually have the two species come back together and form one species again instead of being diverged, because the only difference between these populations is their color patterns and their preference for those colors. So other examples, so some plant species have their own origins in accidents during cell division that result in extra sets of chromosomes. This is what we call polyploidy. So in some plant species, we have issues called non-disjunction during meiosis. And so you'll have some individuals that end up with extra sets of chromosomes. Some plants will self-fertilize and actually create offspring using their own mutated genetics that is completely different from the parent, because they were able to create a new version of themselves through duplicating their chromosomes. And this is called autopolyploidy, where you have the contribution to a hybrid comes from one species, where you don't have two species coming together to create a hybrid species. You have one species that is creating a hybrid of itself that has created an entire new species from its own DNA, in this case through doubling its chromosomes because of a non-disjunction event. We can also have evolution by changes in the timing of growth and development. This can happen through allometry, heterochrony, or pedomorphosis. So allometry is where we have a change in shape or during growth due to differential growth rates. Heterochrony is where we have evolutionary change in the rate or timing of development. And pedomorphosis is the retention of juvenile features in the adult or reproductively mature stage. And we're going to talk a little bit more in detail about each of these. So a good example of differential growth rates occurs in humans. So if we think about the proportions of a newborn baby's head is the largest part of its body comparatively. A newborn's head is a fourth of its body size. By the time you get to an adult, you have your head is about an eighth of your body size. And so your limbs are growing quite a bit faster than your head and your trunk during development. We can see differences in growth rate of different body features when we compare human and chimpanzee skull development. So when humans and chimpanzees are fetuses, their head shapes are very, very similar. Over time, we see that the back of the human skull grows quite a bit faster than the back of the chimpanzee skull, resulting in the differences in appearance between the skulls of chimpanzees and humans. And so that's where we see that allometry, where we have differences in growth and development. Another example of changes in the timing of growth and development occurs in salamanders, where ground-dwelling salamanders have a longer time period for foot growth, and it results in longer digits and less webbing, whereas tree-dwelling salamanders have foot growth that ends sooner, and this evolutionary timing change accounts for the shorter digits and more extensive webbing, which helps the salamander climb vertically on tree branches because they have more ability to stick with their little pads onto the trees. In pedomorphosis, we have the retention of traits in adults that are normally seen in juveniles. So usually what we see is that sexual maturity occurs at the start of adulthood, and those characteristics of sexual maturity do not appear until you are an adult. There's different variations of pedomorphosis. The first one is called neoteny, where body development of the organism is slowed relative to the onset of sexual maturity. And the other is progenesis, where sexual maturity of the organism is sped up relative to body development. And neoteny is much more common than progenesis. We actually see neoteny in axolotls. So this is where they reach sexual maturity without ever undergoing metamorphosis. And so in axolotls, larvae have gills. And so these individuals will maintain gills, and they'll actually have reproductive organs that are sexually mature in their larval stage. And the benefit of that is that they're able to stay underwater and use that environment for reproduction. And so axolotls will be able to reproduce as larvae instead of as adults.